So for those not familiar with the process, let me say just a couple of words before Eithan starts. So Eithan's in the MD PhD program, so he's doing medical school and graduate school all in one mad rush. So for the graduate school, there are a lot of steps to it. He's had to take extra coursework because he's getting his degree in neuroscience imaging, which is part of the medical physics program. So he's been learning the physics of the instrumentation and the mathematics of the image analysis, as well as learning neuroscience. So he's had a pretty heavy course load to address. And after addressing that, then he needs to plan a research project and perform the research project. And the standard for this lab is higher than most. That is, we expect 3 first author publications for people taking a PhD, and most people exceed that. So Eithan's in that group of the top tier of people that are going to exceed that mark. So it's a long haul. He had to propose his dissertation to the committee members, and they gave him pushback and guidance as to what a dissertation should look like to be acceptable. So they approved his work, and now he's completed the course of research that was laid out for him 2 year and a half?
Year and a half ago.
And so he's presenting to you the culmination of all that work. And after he does this, there'll be time for questions from the audience. And then we'll ask you all to leave, and there'll be more questions from the committee. And at that point, Eithan leaves, and then we vote. And so by the end of the day, he should hopefully the final steps, except for turning the forms into the dean's office. The dean's here making sure that Eithan does a good job. And so he should know soon that he'll have completed this phase of his work, and then he gets to go back and resume medical school. So at any rate, so Eithan is here to present to you the final phase of his doctoral degree in a presentation entitled Characterizing the Neural Signature of Metabolic Syndrome. So Eithan, take it away. Ooh.
So Dr. Fox forgot to mention that I also needed to be very well versed in diabetes. So that is some extra coursework that I had to tag along and read about over the course of my project. So as Dr. Fox said, my presentation is Characterizing the Neural Signature of Metabolic Syndrome. But before we begin, I want to ask you guys this question. What makes this Tuesday different from all other Tuesdays? I can see Dr. Weiss chuckling. He knows what that means. Because this is a question we ask in Judaism, specifically in the celebration of Passover. What makes this night different from all other nights? So today marks the 4th day of Passover, and that's why we have matzah and lox and delicious Jewish food. And I think that Passover is a very apt time to defend because it's when we celebrate being released from bondage. And so I empathize with the Hebrews in this case, and I feel like this kinship with 2,500 years ago. So with that being said, I want to begin at the beginning. First of all, thank you all for coming. It's an honor and a pleasure, and I feel incredibly, just amazed that you all made time to come, and it means a lot to me. But to begin, my interest in this subject began when I was in medical school, and I participated in an interdisciplinary organization known as Frontera de Salud. And what we do in this organization is we go to communities, like low-income communities in San Antonio and on the South Texas border, and we do nutrition consultations, lipid screenings, medical education on vaccines, and a bunch of other things. And so one of the biggest issues, obviously, that we see specifically among the Hispanic population in South Texas is diabetes. And in San Antonio, the statistics are rather remarkable. The United States has a prevalence of type 2 diabetes that is diagnosed and confirmed at around 9%. In Texas, it's just a little bit higher, but in Bexar County, where San Antonio lies, it's at 13%. But even within Bexar County, it's much higher among the Hispanic population, almost at twice the national rate. Now, keep in mind that this is diagnosed diabetes. This is not pre-diabetes, which is a much higher rate. And it's also important to note that diabetes is a disease that correlates with aging. It's something that takes many years to develop, and so this is why you see it much more often in individuals over the age of 65 versus, say, younger than 25. Although even in this case, all of these rates are growing in the nation, which is somewhat troubling. And so because I was going to enter Dr. Fox's lab, and because this is neuroimaging, I was interested in, well, what about the brain? What does diabetes do to the brain? And with that, I started doing some literature review, and I was stumbling on a lot of papers such as these: Alzheimer's disease and type 2 diabetes, a critical assessment of shared pathological traits. You see insulin in the nervous system, the role of type 2 diabetes in neurodegeneration, how does insulin resistance develop in Alzheimer's disease. So there's a lot of information out there relating type 2 diabetes and Alzheimer's disease for some reason. And I was very interested in that. And I was actually wondering, well, what is it about Alzheimer's disease and diabetes that is causing this to happen, or is there really such a connection? And so while looking at what was the original study that came up with this hypothesis, I stumbled onto this one, which is the Rotterdam study. And it's a study that takes place in the Netherlands. The Netherlands has a very efficient network for physicians, and there's vast amounts of patient data. And they did a longitudinal study in where they assessed the prevalence of type 2 diabetes over the course of several years, whether or not it led to dementia. And the research concluded that diabetes doubles the risk of developing Alzheimer's disease. So that in and of itself is pretty interesting. There must be something going on. And shortly thereafter, there was a hypothesis based on some animal studies that they found insulin resistance in the nervous system, specifically in the hippocampus. Like, oh, well, if Alzheimer's disease, this is where I want to point this out. So there's a spotlight, right? And Brian would know this one. There's a spotlight in the research where you're seeing this correlation between type 2 diabetes and an increased prevalence of developing Alzheimer's disease. And so what you want to look for are things that cause Alzheimer's disease. So there's a spotlight specifically on this. The one region to be distinguished in Alzheimer's disease, the one that degenerates first before any other, is the hippocampus, or the medial temporal lobe structures. And so this is the hippocampus. The reason why there's a little seahorse is because the Greek hippocampus means seahorse, and that's the hippocampus when dissected out of a brain. So that's why it got the name. And in the 3D image, that's where it is. So that gave me a great idea. And I approached Dr. Fox and asked the question, well, can we use primary data to show that non-demented people with diabetes exhibit a pattern of neurodegeneration that mirrors that of Alzheimer's disease or other known dementias? This is before the onset of Alzheimer's disease. Since Alzheimer's isn't something that just starts in the middle of nowhere, it's something that progresses over many years, and you can see changes in the brain before anyone develops any symptoms. So I wanted to ask the question, well, could type 2 diabetes lead to something like that? Okay, so in the Fox lab, in the Fox lab, there is every single student of his will know the pattern of what we have to do. We come up with a hypothesis, we do a meta-analysis using the BrainMap software that was developed here at the Research Imaging Institute, and we apply that to primary data. Now, the great thing about meta-analysis is that we can use a coordinate-based system because the brain is in 3D space, and there are different coordinates. If you look at an XY axis, there's also a Z axis. And different points in the brain, you can combine a bunch of studies, weigh them accordingly in terms of how many individuals participate in the study, the statistical significance, etc., and create a robust model that can show you exactly which regions are the most significant in any given disease. Unfortunately, when it comes to type 2 diabetes, there weren't enough studies to perform a statistically powerful meta-analysis. So we came up with a different idea. And this leads to my first aim, which is the hippocampal network model, a transdiagnostic metaconnectomic approach. The idea here was that if the hypothesis of Alzheimer's disease connected to type 2 diabetes is correct, you would see a connection between the hippocampus and other regions of the brain. And so if we look at all diseases that implicate the hippocampus, perhaps we can define a specific model that we can then use to apply to primary data. So these are the hypotheses for AIM-1. We can use the BrainMap's disease-based anatomical MRI database to derive a hippocampal network model using a transdiagnostic metaconnectomic meta-analytic approach. Our second hypothesis, that we can use meta-analytic connectivity modeling to assess regional connectivity from BrainMap structural and functional databases, and I will go over that in a bit as well. And we can test the network degeneration hypothesis by identifying network correlations between both structural and functional networks. Now, just as a quick background of what the network degeneration hypothesis is, it predicts that syndromic atrophy patterns recapitulate healthy functional network architecture. And the idea here is that in this case, for example, we see structural covariance in healthy subjects. So if you were to seed a specific region of the brain, you would see a correlation with healthiness in different regions, in gray matter, across many individuals' brains. Similarly, in functional connectivity, you will be able to see the same patterns in the same regions. And going along with that, in the disease parameters, and people who have certain diseases, you will see atrophy in the exact same network pattern. So that's the idea of the network degeneration hypothesis, that structure and function are correlated with one another. Okay, so with our first hypothesis, we wanted to use the hippocampus as a seed. Essentially, it's a seed-based analysis, and what we did is we put the hippocampus, we added the hippocampus and seeded it in the BrainMap database, specifically the functional database that contains all structural, anatomical, disease-based analyses. And from there, we derived a set of regions that basically what this means is that when the hippocampus is atrophied, when the hippocampus is damaged, these other regions are also damaged with the hippocampus. And so this uses something called an anatomical likelihood. And essentially, not to go into a lot of detail in this, this is what I mentioned, remember, with the 3D coordinate system. Well, in every coordinate, let's say this is just one dimension instead of three, there's a point whereby different studies can then pool the coordinates together, give it a Gaussian distribution based on its weighting, and eventually come up with an image that looks kind of like this. But then you add some statistical filtering, and in our case, in the hippocampal network model, we used a family-wise error correction of 0.01, which is highly significant, and we ended up with something that looks somewhat like this. So these are the regions, and I am pointing at the peak regions of coalescence of all these studies. And you see some regions that are interesting. So we see both the anterior and the posterior hippocampi on both sides. We see the right and left claustrum. We see the right and left caudate, the right and left medial dorsal nucleus of the thalamus, the insula, and the claustrum. I forgot that. But in any case, so we have this, right? And so that is our seed-based analysis. And in order to probe it a little bit more, to run a meta-analytic connectivity model, we take each of the foci and create them into spheres. So it looks like that. Now we can analyze or start processing our second hypothesis. And that is using meta-analytic connectivity modeling, or MACM, to assess the interregional connectivity of these specific regions. And it is a little bit of a pipeline, and I'll go over it briefly. And so here we have each of our little nodes, right? 1, 2, 3, 4, 5. There's 11 in total. And we do exactly the same thing. It's also a seed-based analysis. We input each individual sphere back into the structural database of BrainMap. And from here, we get an output of a multitude of studies that basically show that wherever this region is atrophied, to what degree are every other region atrophied. And so we have the anatomical likelihood estimates, and this is what we obtain. And so when we seed this little one right here, we can get up here what's an AO value, an anatomical estimate value. And further, we can also identify, based on a similar model, it's almost this is almost like a negative of the AO value, but we get an uncorrected p-value of significance for each node relative to each other node. So we do that with the AO values, we get that with the uncorrected p-values, and we can assess the significance of the connectivity in a matrix that looks somewhat like this. Once we Bonferroni correct each of the nodes, because there's 11 nodes, we Bonferroni correct so the p-value is less than 0.0001, we are able then to develop a model that looks somewhat like this. And here you're seeing the predictive value, for example, of one node being able to, basically what this means, when you see atrophy in one node, how well does it predict atrophy in one of its connected nodes? And so some are bidirectional, which are the thick lines, and some are unidirectional, such as this. So basically what this means is atrophy in the right claustrum would predict atrophy in the left anterior hippocampus, but the inverse is not as statistically significant, and so we're only using that threshold in this case. Now, we ran a brain map disease inference function that basically uses the meta-analytic data, all derived from the functional database, to see, well, where are these networks coming from? Where do these regions weigh most heavily in which diseases? And this is what we find. That Alzheimer's disease, naturally, it's the hippocampus. Alzheimer's disease is very much connected to the hippocampus, is very much affected, as is temporal lobe epilepsy, which, of course, the temporal lobe is where the hippocampus lies. Mild cognitive impairment, which some people consider pre-Alzheimer's disease, and schizophrenia. Okay, so we did the structure. Now let's look at the function. So using the exact same seeds, we then did exactly the same thing. This time, it's not a diseased database. This is a healthy database of functional studies. A functional study is when you're in an MRI and you're asked to play a little game, essentially. And so there's little games that have to do with a lot of different kinds of tasks, and there's a lot of analyses that have been done, and we've all compiled them, or not all of them, but a good chunk of them in the BrainMap database. So what do these load on exactly? And so we, instead of doing a disease inference function, now we do a behavioral and paradigm inference function. And this is what we find. That these regions correlate most strongly with somesthesis or pain perception, language, explicit memory, attention, other cognition, which includes things like reasoning, emotion fear, emotion other, which includes things like valence, and you'll see a little more of that later on. Now, paradigm classes, these are essentially like the games that people are tasked to play in the MRI scanner. And this is what we see, that reward task is very highly correlated with these regions. So we see here's a z-score of 8, which is highly significant. We also see pain monitor discrimination loading heavily on this network, pain monitor discrimination, passive viewing, and semantic monitor discrimination. And again, we do the exact same kind of matrix. We have another web, another network, and now we can use these and compare them to each other. Now, just by eyeballing it, you can tell that there's some differences and some similarities. Here's another eyeball. You can see that some are definitely the same and others are definitely different. And that leads us to our third hypothesis. And we can test the network degeneration hypothesis by identifying the correlations between the two. And so we ran 2 simple correlation tests just based on the matrices of the AO values, and this is what we derive. We found that using the Mantle test was something that the reviewers wanted to see, and then we did a Pearson's correlation. And we see that there's an R of basically 0.37, 0.4 or so, and an R squared of around 0.15. So what this indicates is that 15% of the variance of the overlap in these networks is related to like structure versus function. So structure is responsible for 15% of the variance associated with function and vice versa. And that's the last of these hypotheses. So take-home messages from AIM-1. So we derived a structural and transdiagnostic hippocampal network model comprised of 11 nodes using the BrainMap's VBM database. We derived their structural and functional interconnectivity profile that we can later apply to primary data. And we found a moderate but statistically significant correlation between structure and functional models supporting the network degeneration hypothesis. Now, I am proud to say that this AIM-1 was published January of last year. And so that's where it is. Yeah. And check it out, Neuroimage Clinical. Okay. Now, before we move on, I just want to give you a factoid about Passover. And in Passover, at every quarter, you sit at the Seder, you sit at the table, and you're telling the story of Passover, of how the Jews were enslaved in Egypt. And at every quarter of the story, you take a sip of wine. So there is no wine available, I'm sorry, so please take a sip of whatever drink you have in front of you. And enjoy. Thank you for those of you who did. Okay, so now we got excited, right? We have the network model. We're going to apply it to primary data. And I approached Dr. Fox, and it's like, great, we have something. This is amazing. What is the primary data that we have? And amazingly, as if almost serendipitous, we have something called the Genetics of Brain Structure Image Archive housed here at the Research Imaging Institute. The goal of this archive is to identify, localize, and characterize genes and quantitative trait loci associated with variants of brain structure and function. Essentially, it's a mass of data that includes different kinds of MRI scans. There's T1-weighted MRI scans, DTI, resting state, etc. We also have genetic profiles, lipid profiles, like blood chemistries, you name it, and psychometric tests. It's comprised of an extended pedigree of Mexican Americans from the greater San Antonio area, and at the time I ran these analyses, there were 1,911 individuals in the database. And of course, Mexican Americans from the greater San Antonio area. This is what comes to mind. So these are the patients that we're looking at from our community right here in San Antonio. When I was sifting through the data, though, I noticed that, of course, those who had type 2 diabetes, let me preempt that with, we wanted to test not just the hippocampal network model, but the next step in our hypothesis was to derive, well, what other regions, what other nodes could be present in individuals with type 2 diabetes. And so for that, we needed to do a contrast analysis. And for that, you need to have a specific grouping of individuals that are age and sex matched, et cetera. And it was very difficult to match individuals with type 2 diabetes versus those without, because those who had type 2 diabetes were relatively old, those who were healthy were relatively young, and so the grouping didn't really match too well. So that's when I approached Dr. DeFronzo. And we talked about, well, the status of what prediabetes entails and insulin resistance. And it turns out that using insulin resistance as a marker to group individuals is a pretty decent idea, because in terms of years, it takes a long time to develop type 2 diabetes in the general population. But what we see is that over the course of years, there's a spike in exogenous insulin or chronically elevated insulin. There's also an increase in insulin resistance in individuals that basically correlates very strongly, specifically with central obesity. And at one point, you start getting impaired glucose tolerance, and eventually your beta cells that produce the insulin in your pancreas start dying, and you end up getting increased blood glucose, which is characteristic of type 2 diabetes. Now, Dr. Fox showed me this, which was a white paper published by the National Institute of Mental Health that was very concerned in looking at the mechanisms between neurocognitive dysfunction, insulin resistance, and other metabolic diseases. So and it gave a whole list of propositions, like this is the research that could be done, and I took that to heart. And this is its summary figure that I found very interesting. So we have metabolic syndrome, and we see that there's insulin resistance, there's obesity, prediabetes, diabetes, they relate to one another. And at the same time, there's neurocognitive decline in dementia. And citing the Rotterdam study, as well as other replicated studies, looking at the association with dementia and type 2 diabetes, this is an interesting connection that we can explore still further. And so instead of doing the analysis between type 2 diabetes and non-type 2 diabetics, I figured, well, insulin resistance is probably the next best thing, because obviously it comes before type 2 diabetes. So pre-diabetes, pre-dementia should somewhat load. That was my thoughts on that. So there's a twofold increase in developing dementia, all-cause, and Alzheimer's disease. The white paper also mentions cerebrovascular disease, also vascular dementia, and accelerated brain aging. Now, there's also some impaired cognitive functions in metabolic syndrome, including things like impaired executive function, learning and memory, and processing speed. dysregulation of emotional affect, especially in young people. There's a huge incidence of depression and anxiety in individuals with type 2 diabetes, especially young individuals. There's also dysregulation in reward perception, so the appetitive network involvement, which is basically the reward network plus the hypothalamus that regulates satiety and other things. And so because we don't have the metrics for insulin in each of these individuals, it's like the one blood chemistry thing that we were missing, we defined our samples using metabolic syndrome. And metabolic syndrome, in short, is a combination of 3 of these 5 factors, according to the International Diabetes Federation, the National Cholesterol Education Program. So we have central obesity as 1, raised triglycerides, lowered HDL cholesterol, which is the good cholesterol, raised blood pressure, and raised fasting plasma glucose. Now, one epidemiological study showed that metabolic syndrome, as with diabetes, is a disease of aging, but it loads differently between sexes and between individuals of different ethnicities. And because we have the genetics of brain structure dataset, because we're concerned about the community in San Antonio, we are looking at the individuals who are of Hispanic origin, specifically Mexican Americans, who are more highly predisposed to developing metabolic syndrome and all of its comorbidities, including cardiovascular disease, diabetes, etc. But this is the main message. 34.7% of the US population, adult population over the age of 20, qualify for a diagnosis of metabolic syndrome according to the International Diabetes Federation. And so that is concerning, to say the least. So let's begin now. Because of this, we're going to characterize a neural signature of metabolic syndrome, our second aim. And first, we hypothesized that we can use primary data to perform a contrast analysis, as I mentioned, revealing metabolic syndrome-specific gray matter volume differences. Then, we tested whether the patterns of gray matter atrophy observed in metabolic syndrome will recapitulate regions of known dementias like Alzheimer's disease and mild cognitive impairment. And finally, we wanted to apply the meta-analytic interpretive tools that I mentioned earlier to these structures that were derived to identify cognitive domains corresponding to the cognitive deficits that were cited in this white paper. So again, let's use primary data, contrast, gray matter volume differences. And we use the genetics of brain structure. And the idea here is to identify individuals, match them precisely by age and sex, so they're perfectly controlled for age and sex, of the healthy spectrum and the metabolic syndrome spectrum. So we scored each individual, however many metabolic syndrome criteria they had, and we matched them with those who had very few. And yes, metabolic syndrome, there we go. And so this is basically what we did. We had 1,911 individuals from GOBS. We then removed all the individuals who did not have adequate MRI scans, like those MRI scans that were incomplete, and even those who did not have matching lipid panels, because we need lipid panels to assess whether or not someone has metabolic syndrome. And from there, we removed everyone who had a past history of stroke, neurosurgery, neurological disease, and Axis 1 psychiatric disease, according to the DSM. And from here, we then defined, well, what is someone who is healthy, someone who's metabolic syndrome. And essentially, we overshot a little bit with metabolic syndrome. We wanted someone to be definitely have metabolic syndrome. So have 4 or 3 of the 4 criteria and definitely have increased in central obesity. Similarly, in healthy controls, we were a little bit forgiving. We allowed 4 out of 5 criteria to not be present. So someone could have maybe a blood pressure that's a little bit high, so long as they didn't have any of the other criteria. They were included in the healthy group of individuals. And by the end, we had 104 individuals on either side, which in and of itself is the most powerful, largest study of its kind looking at metabolic syndrome. Others, having looked at diabetes and obesity, are a little bit larger, but in terms of metabolic syndrome, this is really the largest that has been done. I think the second one was groups of like 15 individuals, not groups of 104. So we used something called voxel-based morphometry to compare metabolic syndrome versus metabolically healthy. And what this is, it's a 2-group difference, 2-sampled unpaired t-test using voxel-wise general linear modeling. And this is how it begins. We first get the raw images from the individuals. These are T1 weighted images from our T3 scanner, which is a very fancy scanner. Then we extract the brain, so we scalp it. It's the best term to use. We remove the scalp, we remove all the other non-brain tissue. From there, we segment it. So there's different brain tissues. There's the white matter, cerebrospinal fluid, and gray matter. And so we're only interested in gray matter for this particular study. So we remove the gray matter. Then we create a study-specific template based on the standardized MNI template right there. From there, we then individualize. So each individual brain gets standardized to the study-specific template. And from here, we modulate and smooth it so that it's easy to analyze. And we do a comparison. And essentially, this is a simplified version of what the study template looks like. There's control greater than metabolic syndrome and metabolic syndrome versus control. And so we want to look at all of the parameters with regards to this. So it's important to mention these are adjusted for age and sex. Okay. So the main effects of our VBM contrast analysis between 104 metabolically healthy and 104 individuals with metabolic syndrome. So we have a mean age of 37.3 years. Another important thing to mention is that the other studies tended to look at individuals who are older, over the age of 65. So this is also the youngest cohort that has been studied in the literature using structural VBM analyses. We have a total of 208 individuals in our study. And we family-wise error correct at a P of 0.05 at 10,000 permutations, which basically is a way to eliminate as many false positives as possible. And this is the end result. Take a few seconds. This is my favorite figure. So this is the neural signature of metabolic syndrome. And it was very surprising that we saw this big of an effect. We were expecting maybe just a few little nodes that we could add to the hippocampal network model, but it turns out it's its own thing by itself. So one of the sayings that I remember hearing is that science doesn't advance necessarily by testing your hypothesis and advancing perfectly. Sometimes it's like, that's funny. So that's how science really advances. And so that's what we found with this analysis. And for the anatomist aficionados, here are the regions. We have the right superior temporal gyrus, the right insula, the right amygdala, the caudate nuclei, and we have the orbitofrontal cortex, or the ventromedial prefrontal cortex, the lingual gyrus, the brainstem, and the posterior cerebellum. That's the most interesting one. Red is more significant, by the way. It's less than a P of 0.001. Okay. So I want you to notice something. Where's the hippocampus? There's absolutely no overlap. I'll show that to you again. Red is a metabolic syndrome. Green is a hippocampus. No overlap whatsoever. So, is it Alzheimer's disease or something else? Okay. So our second hypothesis, we wanted to see, well, what do these regions... We're now looking, just by virtue of looking and eyeballing, well, it's not the hippocampus, so could it be Alzheimer's? We wanted to still do the same disease inference function. But because we were looking at individuals who were 37 years old on average, I mean, it's a range of between 18 and 72 or something, we wanted to do the same thing, but instead we divided them up by the median age. And so we had two half samples, one who was young and one which was young and one which was old. And there we go. So now we have a healthy control. So if we do the same analysis using the half sample of the younger individuals, average age 26, we see that the posterior cerebellum is the only region that is atrophied, or that has a lower gray matter volume. If we look at individuals who are older, average age 48, I'm sorry, but there's you see basically the same pattern as the whole sample put together, but it's a little bit more diffuse. Still, though, the hippocampus isn't overlapping anywhere. Yes. Okay. And so this is what the disease inference function looks like in the full sample. What we see is, for our purposes, this kind of analysis is considered statistically significant or worth mentioning if it has a z-score of greater than 3. And we see Alzheimer's barely meeting that threshold, as well as Huntington's disease barely exceeding it. And my hypothesis with this is that because the caudate nucleus is very prominent in Alzheimer's disease individuals, that's probably what's weighing Huntington's disease a little bit more. In terms of Alzheimer's disease, that was a little bit of a mystery, but we do see the same regions from the hippocampal network model. We still see the caudate, we still see the insula, So there still could be some remnant of the hippocampal network minus the hippocampus, which is probably why the Alzheimer's is almost at that threshold. Looking at the older half sample, we see that Alzheimer's is a lot more significant now, and because it's much more diffuse. It's using more of the insula, which is prominent in Alzheimer's. It has the other side of the insula as well, and again, it's Huntington's disease and schizophrenia. Now, for our third hypothesis here, I mentioned earlier the different kinds of cognitive deficits experienced by individuals with type 2 diabetes and metabolic syndrome. And so we wanted to do the exact same inference function like we did earlier, this time looking at the other regions, or looking at behavior domains instead of disease domains. And so this is what we see, that it is these regions load highly on reasoning skills. So this is part of executive function. It's a subdomain of executive function and cognition. And emotional valence, which is the ability to perceive and express the range of emotions. Similarly, in the paradigm analysis, we see the reward network also very highly significant. Looking at the older half sample, we see basically the exact same regions, or the same domains. Emotional valence and reasoning are very high. Maybe fear, music, audition are a little bit lower in this case. And with the paradigm analysis, we see reward, face monitor discrimination, pitch monitor, and music comprehension. Okay, so take-home messages from AIM 2. That a neural signature of metabolic syndrome exists, and it is distinct from known neurodegenerative diseases. It wasn't as highly significant to Alzheimer's or Huntington's as we would have expected it to be to any other disease. So there might be something unique in metabolic syndrome, or at least in dementia, or dementia that is related to metabolic syndrome and diabetes. Though slightly similar to Alzheimer's disease, the absence of the hippocampal involvement suggests that pathogenesis is not that of Alzheimer's, because the pathogenesis of Alzheimer's begins in the hippocampus. And if it's not beginning in the hippocampus, then I would say it's not Alzheimer's. It's similar to some degree, but not exactly the same thing. Regions implicated in the neural signature of metabolic syndrome are regions responsible for reasoning, emotional valence, and reward perception. Also, we do note that there are age-related effects. Those who are younger experience atrophy in the posterior cerebellum. Those who are older, it's a more diffuse pattern. And the most significantly affected region is the posterior cerebellum. You may have your second glass of wine. Okay. AIM 3. Does cognitive decline in metabolic syndrome begin with the posterior cerebellum? We know that neurodegeneration begins in the posterior cerebellum, but does cognitive decline itself do as well? And so we hypothesize that neuropsychological effects pertaining to metabolic syndrome correspond to similar effects pertaining to posterior cerebellar damage. Now, for many of you, it's been inculcated that the cerebellum has to do with movement. If the cerebellum is damaged, then your movement is impaired, your coordination is impaired, etc. And so this gentleman, Marie-Jean-Pierre Florenz, was the first scientist to demonstrate experimentally that the cerebellum is responsible for equilibrium and motor coordination in the 1800s. He did his experiments on rabbits and cats and dogs, but not on people. But nevertheless, between the 1800s and the 1900s, there were clinical cases of people who were missing the cerebellum or who had strokes in the cerebellum that were off. Like there were some clinically relevant problems in their emotional perception, in their cognition or something. And then this gentleman, Jeremy Schmahmann, who's a Harvard neurologist, he specializes in the cerebellum. He wrote the textbook on the cerebellum. He was the first to clinically and experimentally establish the presence of non-motor symptoms of cerebellar dysfunction. And this was his first paper in 1991, I believe, an emerging concept, the cerebellar contribution to higher function. So one thing to note is that the posterior cerebellum, as opposed to the anterior cerebellum has to do with movement. However, the posterior is also known as the neocerebellum, just as we as humans have a neocortex, which is the prefrontal cortex that allows us to do higher cognitive functions. We also have a neocerebellum that modulates these higher cognitive functions. And so he called this the dysmetria of thought. In a typical cerebellum experiment, you put your finger out and touch your nose, right? So this is establishing coordination. And if it's staggered and you can't aim right, there might be something wrong with the cerebellum. So that's dysmetria. What about dysmetria of thought? How can you even assess that? And so he looked at 70 patients, he assessed their syndromes and everything, and established something known as the cerebellar cognitive affective syndrome, which one of his colleagues eventually ended up naming Schmahmann syndrome. Now, some characteristic deficits of Schmahmann syndrome, or posterior cerebellar damage and degeneration, are the following. There's problems in executive function, specifically in planning, abstract reasoning, multitasking, and working memory. Spatial cognition, impaired visuospatial memory. Deficits in language, which include dysprosody, dysprosody is basically deficit in intonation, and even understanding things like sarcasm or questions that require language intonation. There's agrammatism, which is telegraphic speech, so having fewer words that are descriptive, and anomic aphasia, which is like when you're at a loss for words. Okay. Deficits that affect regulation, deficits in affect regulation and perception include things like blunting of affect, disinhibition, and inappropriate behavior. Now, if we take the metabolic syndrome, the neural signature of metabolic syndrome, and make it super statistically significant, we look at the FWE value of 0.0001, and so definitely there are no false positives at all in these regions. This is the anatomical region that we get. And so this is what we wanted to study relative to psychometric tests. Now, last year, coincidentally, the cerebellar cognitive affective syndrome has a scale was published for this syndrome. And it's a way to assess the degree of severity for individuals who experience a tumor resection in the posterior cerebellar region or a stroke or anything like that. So this is now well established and valid. And we wanted to use it, but we don't have the tests, or do we? We have the South Texas Assessment of Neurocognition that was given to each one of the GOBS patients that we had in our study. And these include a lot of overlapping psychometric tests. And here we can see that we can match a lot of these with the Schmahmann scale. Unfortunately, we can't match all of them, so some had to be removed. So what we wanted to do in this case was to assess whether or not there is a correlation between cerebellar volume, posterior cerebellar volume, and lower scores. Now, one of the things I want to point here is that the mean age of the individuals are, it should be 26.5, but it's the younger individuals from the original cohort. We wanted to use younger individuals because older individuals, we don't know how long they've had metabolic syndrome, if they were diagnosed recently, but at least we know that those who are young should not have it at all. And so we can do a psychometric analysis more effectively in individuals who are young with metabolic syndrome versus individuals who are older with metabolic syndrome. And this is what we see. We recapitulate the findings of the posterior cerebellar gray matter volume, where there's a decrease in cerebellar volume. These are metabolic syndrome. Those are the healthy controls. So there's a relatively large effect size, significant difference between the two. In terms of white matter, we also see a difference, p of less than 0.001. I also wanted to add the hippocampus just for posterity, because everyone kept talking about the hippocampus in the literature. Now I'm not convinced it's a hippocampus at all. But here we can see that there are no differences in hippocampal volume. As a control, if you want to look at brain regional differences, we also want to have the whole brain gray matter volume. We don't want to say, oh, well, there's a decrease in posterior cerebellar volume, but it is also corresponding with whole brain. We want to see that there's a difference. So there's no difference in whole brain gray matter volume between these two groups. Now, if we look at those who had our adapted Schmahmann scale, it's a lot more interesting than we expected, because even though it's not a p-value of 0.05 or less, it's a p-value of 0.06, which is a trend. And consider that we don't have the perfect Schmahmann scale applied to each of these patients, and it's adapted, but at the same time, we are seeing a relationship between the cognitive scale based on the Schmahmann syndrome and as compared to healthy individuals. Now, this is a composite of the adapted Schmahmann scale, and here we, as a comparison, we combined the entirety of the South Texas assessment of neurocognition and found that the p-value is 0.09. So it's still there. You can still see a difference. It's not statistically significant, but just as an observation, this was interesting. So our take-home messages from this analysis is that the posterior cerebellar degeneration observed in young people with metabolic syndrome likely reflects neurocognitive changes consistent with Schmahmann syndrome. Preliminary data, it's interesting. It's not yet conclusive, but it certainly gives us ammunition to maybe pursue this a lot further in a future study. We were limited by the sample size because we were only looking at 104 individuals, and the effect size, which we had an effect size of a Cohen's d of 0.3, which is like a third of a standard deviation. So it's there, but it's not as convincing just yet. If we had a larger sample size, if we had the right neurocognitive assessment, then that would have been a lot more exciting. Third glass of wine. We're almost there. Okay. So aim 4 is the aim that we wanted to do initially. We wanted to apply the hippocampal network model into the general population and assess all these different connectivities. But because the hippocampal network model got by the wayside, because the hippocampus was definitely not involved in metabolic syndrome, and because metabolic syndrome was so interesting in and of itself, or that at least the findings were that interesting, so our first hypothesis in this case was the effect of each metabolic syndrome factor on the neural signature of metabolic syndrome. Brain structures will identify discrete differences between the sexes. So we noticed in the original epidemiological study that there were sex differences, and I'll go over that a little bit later. Our second hypothesis is basically the same thing, but also differences between age groups. And our third hypothesis is that the gray matter volume in the neural signature of metabolic syndrome regions exhibit a linear relationship with metabolic syndrome factors in a representative population, implying that the relationship between gray matter volume in these regions and individuals with metabolic syndrome components lie in a continuum. So it's not either you have metabolic syndrome or you don't. It's that it's a continuous thing that loads perfectly in terms of gray matter volume differences and increased metabolic syndrome factors. So some noticeable differences between the sexes in terms of metabolic syndrome. We know women live longer than men. And so that has to do with a hormonal profile, and that affects the metabolism and metabolic function of the organism. Also, men tend to exhibit higher levels of triglycerides and blood pressure, but lower age-adjusted levels of waist circumference and HDL compared to women. There's also the case that premenopausal women more frequently develop peripheral obesity, which is healthier, with subcutaneous fat accumulation, whereas men and postmenopausal women are more likely to develop central, or what's called android obesity, so in the viscera, surrounding the organs, basically just surrounding the belly. It's much more common in men and in postmenopausal women. Also, men were seen to perform worse on memory and executive function tests relative to increasing metabolic syndrome factors as compared to women. So we know that there are differences. And based on the population that we have, what are they? And just as a recap, you can see that there are definitely age differences as well. So someone who is in their 60s has a higher likelihood of developing metabolic syndrome than someone in their 20s. It takes time to have that increased insulin resistance, chronically elevated insulin, for it to actually cause the damage into the metabolism and all these other things. So what's the best way to divide people by age? It's incredibly arbitrary. So I Wikipedia'd age differences and found one that I liked. And so we have the total subjects and gobs. We have the 835 that have the completed MRI scans and lipid panels. And we excluded individuals with no past medical history of stroke, neurosurgery, neurological disease. Okay. And we have a group of individuals who are early adult, so men ages 18 to 25. Women, early adult women, 18 to 25. Young adult men, 25 to 45. Young adult women, 25 to 45. Middle-aged men, 46 to 65. And middle-aged women, 46 to 65. Unfortunately, we did not have enough individuals to constitute a robust sample size for older than 65. If you were interested in that, I'm sorry. Also, very importantly, these are population-based participants. This is not disease-based. I'm not looking to see whether or not they have metabolic syndrome and compare them to healthy. This is basically the normal distribution of each of these factors. So those who are healthy versus those who are unhealthy, it's not very clear-cut. It lies on a continuum. We did a one-way ANOVA to compare the sex differences, controlling for age, and we see a few interesting things. So the most significant differences between the sexes, most of these are significantly different, but the biggest differences are in triglycerides, HDL cholesterol, and blood pressure. Not different, which was also interesting, was the rate of type 2 diabetes diagnosis of hypertension, diagnosis of hypertriglycerides, and hypercholesterolemia. Now, if we do another one-way ANOVA, this time just within each of the sexes, comparing the age groups, you would expect to see a huge difference in each one of them, but at least in men, HDL cholesterol was not significant, and in women, HDL cholesterol between the sexes was just barely significant. So this is another way of looking at the neural signature of metabolic syndrome. And what we wanted to do is we wanted to use each of these regions as independent variables in a regression analysis, incorporating all of the metabolic syndrome factors. So what we did to extract these gray matter volumes is we have each of the GOBS participants. This is already the process brain. We have a region of interest, and it's surrounding regions that have gray matter signal, essentially. And this is a normalized gray matter volume, also known as a gray matter density. And what this is it's volume divided by the signal, white meaning signal, black meaning no signal. And we did that with each individual. And this is another table demographic that we want to show. So again, another ANOVA looking at the volumes of each of these regions. We have the whole neural signature. We have the whole brain as interests that we also want to look at. Also the hippocampus, again, for posterity to really drive the point home that it's not the hippocampus. And we see that there are certain differences between the sexes. Specifically, what's interesting, posterior cerebellum and hippocampus were no different between men and women. These are the two structures that did not differ in size between men and women. Okay, so our first hypothesis, are there differences between the sexes? So this is another table. It's a little prettier than the earlier ones. But what we see here is that the degree of convergence between the metabolic syndrome structures and the metabolic syndrome factors are weighted differently between men and women. And I want to focus just on these. These are just the metabolic syndrome factors by themselves. These are some other factors that are interesting, but I don't have enough time to mention them. And so we see, again, the brain regions and metabolic syndrome factors in men and women, and we see that the most significant regions are in terms of waist circumference. So waist circumference and the posterior cerebellum are very highly correlated and very significant, as is the brainstem and the entire neural signature of metabolic syndrome, all of the whole model together. Interestingly, it's more significant in men than in women. This is, of course, controlled for age. And what I found interesting is that fasting plasma glucose basically has no effect on at least brain structure in men, but it certainly has a high degree of significance in women. It's not the correlation isn't very high, but it is significant. So, again, hippocampus, nothing to look at there. Okay, so let's just focus on these because there's a lot of information in these tables. What about the differences between the age groups? So here we did a univariate linear regression analysis looking at each individual brain region as an independent variable and all of the metabolic syndrome factors as dependent variables. And we see that in the neural signature of metabolic syndrome, waist circumference and triglycerides, this is in, these are the early adult men, 18 to 25. So the waist circumference and triglycerides loads most heavily on the whole neural signature and the posterior cerebellum at an incredibly high R squared. So this is very, very correlated with, basically what this is saying is that waist circumference and triglycerides are the biggest predictor of gray matter volume in these regions in young men or early adult men. So that's very high, much higher than we expected. And in women, it's also higher than we expected, but a little bit lower in women than in men. Still significant, and it's still waist circumference that loads heavily on that. Okay, and so this is our scatter plot, which is just a way to drive the point home, that early adult men have an R squared of 0.35, or should have been 0.39, but women the R squared is less than that of men. Oops. Okay. Okay. So again, I mean, it's the same idea. We see that waist circumference keeps popping up in this is young adult men now, 26 to 45. In women, we see waist circumference again. This time age starts to creep in and be one of the factors that addresses gray matter volume. Again, we have an R squared. In men, in young adult men, the R squared is a little bit lower. In women, it stays around the same. And here in the middle-aged men, we start seeing that age becomes a bigger factor in terms of predicting gray matter volume, all except in the posterior cerebellum, where waist circumference is still significant, even though the R squared is now getting a little bit lower. In women, we start seeing that there's an increase. So middle-aged women, presumably because of menopause, are already experiencing the effects of fasting plasma glucose and waist circumference more heavily in terms of predicting gray matter volume differences or gray matter volume in general in these regions. And here we have the R squared. So in women, it's still relatively high, and in men, it has decreased. Okay. So, take-home messages from this last aim. Sex differences between the relationship of metabolic syndrome components and brain structure exist. The relationship of combined metabolic syndrome factors on gray matter volume within the neural signature of metabolic syndrome is most diffuse in women. But on average, the metabolic syndrome component of waist circumference, which is very highly correlated with insulin resistance in general, is the strongest predictor of gray matter volume in the neural signature of metabolic syndrome and the posterior cerebellum in particular. The predictive value of waist circumference on posterior cerebellar gray matter volume is also most pronounced in young men. Your fourth glass of wine, please. Now, in conclusion, I'm not going to go over these in any detail. I just want to mention that some of the next steps include genetics. We have in the works the plan to run genetic analyses. We have SOLAR, which is a genetic software that we can use. It's imaging genetics. We can use the imaging volumes or the gray matter volumes and input them and compare them to those of waist circumference or other covariates and identify what the pleiotropic nature of this relationship is. We can assess heritability. We can even do a full genomic-wide analysis if we wanted to. That's the idea. It's wishful thinking at this point, but it's in the works. We also want to do some functional analyses. Everything that I presented to you today was structural T1 weighted imaging, but we would like to apply this to functional connectivity, much like we wanted to do with the hippocampal network model, since we addressed the connection between structure and function. Now we want to see function relative to metabolic syndrome. And of course, the whole idea, we could apply it clinically, translational science. Being able to incorporate neuroimaging in clinical trials of diabetes medications or metabolic syndrome medications would be very informative, especially if we know that there could be some negative cognitive impairments. We want to have this as something to think about. So maybe future studies with Dr. DeFronzo's help, I know he's mentioned it before, we could incorporate an imaging protocol into some of his patients so that we can analyze whether or not there are brain differences in individuals receiving certain treatments. Now, before I end, there's something very important I need to mention to you all. So we went over all these different diseases. We have metabolic syndrome, and it's of course associated with vascular dementia, with cardiovascular disease, with diabetes, with renal hypertension. Like there's so many things that can stem from metabolic syndrome or the diseases that I've been studying these past 4 years. But these past 4 years have also been very hard on my family because these diseases have faces, these diseases have names, and these are people very dear to me who've passed away these past 4 years. And so I just want to send you all with a message that even though we are very excited about our data analysis, that we study something that we were looking at very heavily, that we do so for a purpose in the end. And in the end, it's people like my family. So with that in mind, hear, O Israel, for the next 40 years, we will power walk and eat only matzah. Thank you all. Now I will take your hardest questions. Yes, Dr. Weiss.
Circumference in the progression of this disease something that occurs earlier or later in the pathophysiology?
What's interesting is that it sometimes it relates to one's environment and ethnic background. So individuals who are of Hispanic or Native American descent tend to accumulate more visceral fat than those who are of European descent for some reason. I mean, it has to do, the genetics of type 2 diabetes mainly have to do with the function of the beta cells in the pancreas. And so if there's a dysfunction in the beta cells, if the environment, if you're consuming, say, like a lot of sodas as part of your diet, then that could affect the visceral fat. And so, yes, it could occur as early as young, but it tends to be more age-related, I would say. Yes.
Sort of a follow-up. Do you think that the differences that you see between men and women and the correlation or the sort of statistical significance with waist circumference and the brain regions and metabolic syndrome, do you think that has something to do with the hormonal component and adipose tissue and hormones and estrogen?
Yes.
And these things. And then are you maybe not confounded, but is there maybe an association of women and their hormonal whatever, or their menstrual cycle, or their menopausal, has something to do with the fact that maybe the correlations weren't as strong?
Yes, that's why we it was a population-based study, and we only looked at these factors. In probably a further analysis would have to include those as covariates for sure. But we just wanted to look like straight, like these are the ages. Pre and postmenopausal, especially in women, is very like it's a huge factor in terms of hormonal shift in metabolism, of course. Yes. Okay.
I have a question about some of the findings that you mentioned when you just started off, the link between diabetes and the correlation between diabetes and Alzheimer's. Mm-hmm. In your research and in your work, is that stronger the less manageable they are in terms of their medication management? In terms of how they manage their own diabetes, does that correlation become much more significant? Is there any...
Well, there's
because in the community, we know that there's a lot of people that do not manage well.
So the idea here is that the cognitive deficits that you're seeing in individuals who have type 2 diabetes are probably not Alzheimer's disease. That's the take-home message overall, because it's not involving the hippocampus, but it is involving other correlated regions that still lead to dementia. And it probably leads to some confounding of the Rotterdam study, because really to diagnose Alzheimer's disease, oftentimes in the clinic you just do some evaluation and, okay, this person has a profile of Alzheimer's disease, but that may not be what they have. Oftentimes in the South Texas Alzheimer's conference that we went to just a few months ago, they mentioned that someone with frontotemporal dementia manifested as someone who had Alzheimer's disease. To really diagnose Alzheimer's disease, you need a PET scan, you need to see whether or not there is like impaired glucose metabolism in the hippocampus relative, et cetera. So, but to answer your question specifically to the dementia and poorly controlled diabetes, yes, there is research showing that poorly controlled diabetes is worse for like cognitive outcomes. Yeah, exactly. Yes.
So, very nice piece of work, by the way.
Thank you.
Very nice. Could it be that you don't see an overlap with hippocampi in patients that you looked at metabolic syndrome because you particularly selected for a group that were fairly young, right? Mm-hmm. If you say had looked at much older participants, could you really exclude that there's an overlap?
Exactly. Yes, that's a great question. And there is studies, all the studies that have shown, like the Jan Biesels and the Rotterdam people that have studied this more closely, they have only looked at the older individuals. So they only look at people over the age of 70, and yes, those who have type 2 diabetes over the age of 70 do have smaller hippocampi relative to healthy individuals over the age of 70. But the nature of Alzheimer's disease and hippocampal abnormalities means that it should begin with the hippocampus. The fact that it's starting outside and moving in shows that it's not the same pathophysiological process as Alzheimer's, which would start in and move out, if that makes any sense. Yes. It's my mother. Yes.
Okay. Too much for me to understand. So forgive my question. But there is the metabolic syndrome that is related to this
Waist circumference.
And the fact that I asked you, I want to bring some food for you guys. And you said, fine, just no sugar. So sugar makes you have metabolic syndrome?
Well, it's certainly a big factor, especially if you overdo it. I just wanted to be a model of no muffins in the morning. Because lox and fruit are much healthier than a muffin or donuts or things like that. But
it would be interesting. It would be interesting for a fifth grader to know how not to get
metabolic syndrome. Yeah, it's you have to have good cardiovascular health. So exercise is important. You need to eat a proper diet with a good amount of protein and complex carbohydrates that take longer to digest instead of the simple carbohydrates that are present in sodas like high fructose corn syrup or very heavy sugars or very starchy foods. So all of that definitely increases insulin production and leads to chronically elevated insulin and insulin resistance. And eventually, so all of that is connected to one another.
Most importantly, pick your parents smartly.
It
looks like the both of you are okay. Metabolic syndrome.
Yeah, so there's a huge genetic effect, and so that's very true. Yes, Brian.
Can you hypothesize as to what are the major biological differences between the hippocampus and posterior cerebellum that are going to be making one selectively affected in metabolic syndrome versus not the other?
So there are some hypotheses regarding that. What most of the literature said was that there are insulin receptors in the hippocampus, and so insulin resistance in the hippocampus in individuals with Alzheimer's disease is actually a thing. But the fact that you're not seeing degeneration in the hippocampus and the cerebellum makes it interesting. One of the findings from one of the papers that I saw was the presence of leptin receptors in the posterior cerebellum that is not present anywhere else in the brain other than the hypothalamus. So if there's leptin resistance as well, that could be somehow a mechanistic thing that has to do with it, but I have no idea. Yes?
Is there a way to look at vascular changes in the blood supply of those different areas? So for example, to look at the vascular changes, the blood supply to the hippocampi, say for example, relative to the posterior cerebellum, for example.
That's part of the functional analysis that is up and coming. Yeah, it's because it's blood oxygen level dependency. Bold signaling is basically we're measuring blood flow in these regions. And so that would be an interesting thing to find. And
how would you interpret the DTI study that you showed us? That the DTI effects are very diffuse, whereas the gray matter effects are very specific.
Exactly.
Most people take DTI as an indication of small vessel disease. So,
right.
So it isn't small vessel disease, it's something else.
Yeah. How would I interpret the DTI finding?
Yeah.
Well, it could very well be some level of small vessel disease. The DTI findings, I didn't show it here, but the DTI findings in the older individuals showed that there were no differences between healthy individuals older individuals versus those with metabolic syndrome. The only differences were in gray matter. You saw the DTI findings in the younger individuals much more diffusely. I don't really know what to make of it other than there could be some vasculopathies that could be also associated with insulin resistance and that kind of process. So. Yes.
Can you speak a little bit more about the emotion and reward that you found in relation to metabolic syndrome, and also how do you think this pattern would differ for those individuals that would then go on to develop possibly diabetes? Because it was interesting. Obviously.
Yeah. So it's not. What the findings are showing is that these are regions that in healthy individuals function in the reward pathway and in the emotional domain of valence and other things. So it's not a conclusive finding that if you experience metabolic syndrome, then you will have this impairment exactly. But it's something. It's more of a hypothesis generating thing. Like, we see this correlation, let's explore it further, and that's why it's interesting to see like the whole Schmahmann syndrome connection to everything, because Schmahmann syndrome is very specific to a lot of the cognitive findings they've cited in metabolic syndrome and diabetes literature. So that's the only thing I can really say to that, unfortunately. But yeah. Yes.
So again, this excellent study, you've only been able to look at time points. So for example, the MRIs that you have are studied a point in time. If you were to look at the same patients over a period of time, would you predict that there would be differences, and what sorts of differences would you predict?
Yes, I would, because that's right. It's a big limitation that we had, is that it was a cross-sectional study. So one of the, and I think there is a grant in the works to bring back people from the GOB study, like between 5 and 10 years after?
10 years. The study's already begun. There'll be 700 of the oldest GOBS patients we brought back to do a longitudinal gene by age interaction study.
So I would expect to see a pattern similar to the one that we see in the cross-sectional study. That would be my hypothesis. Whether or not it's different on an individual level could be very relevant to the genetic factor associated with it. So it'll still be very interesting, and whenever the 700 people are scanned, whenever that's done.
It'll take another 3.5 years.
Yeah, so I'll be gone, but I would still like to be involved. Maybe with some future MD PhD student, they can take up the project.
Yeah, just one comment on this, that the cerebellar findings were so unexpected. There's nothing in the literature that predicts that. And so nobody's looked there. So nobody does neuropathology of the posterior cerebellum in diabetes. Right? Nobody's done anything in the posterior cerebellum in diabetes. So now we know where to look. And so in Ralph's patients, we need to start looking at pathology when the patients pass. We need to start using this as a probe, right? This is now a way we can look and we can study this. So the term we use a lot for these imaging features that we acquire is they become endophenotypes, that they're cryptic. No one knows, it's not, like, obesity is a phenotype, but these things are hard to see and take a long time to find them, but they're very potent. So we'll be looking specifically for what genes control that spot in the cerebellum and how do they predict metabolic syndrome. And so now we have a lot of tools from this that we can apply over upcoming years. So I'd say that's a big part of this. These are completely new findings, but they have a lot of long-term implications.
It's done. Oh, yeah.